Hey guys, welcome back. Today we're still in Unit 1.3, World of the Cell, but we're going to look at Part 3, which deals with cellular transport. Now what that is transport of molecules into and out of the cell. So let's go ahead and get started. All right, so first thing we have to do is talk about something called concentration. Now concentration, that's not when you think really hard about something, not in this case. Concentration is a term used to describe the mass of the solute per volume of solution. So in other words, how much of a solute is there in the solution, in the actual solvent? Now, the more solute molecules that are present, the higher the concentration. And thus, vice versa, the less solute molecules present, the lower the concentration. When there's a lower concentration, we say it is diluted. So if we look at this picture on the top right, if there's just a little bit of salt in a cup of water, it's a very low concentration of salt. You might not even be able to taste the difference. It's very, very low concentration, so we would say it's a diluted concentration. But if you put a lot of salt in the water, now you have more salt per the same volume of liquid. So we have a much higher concentration. Now in nature favors equilibrium. And what that is when the concentration is equal on both sides of a membrane. And that's what we see in this bottom picture here. So here's our cell. And we notice that the concentration of whatever solute this is much, much, much higher on the outside of the cell. There's more of it on the outside than there is on the inside. So what happens is the solute molecules will actually move from an area of higher concentration to an area of lower concentration. So it's higher on the outside of the cell, lower on the inside. So we're going to see some of the molecules go across the cell membrane and enter the cell. Now when that happens, if you notice in the final picture there, that, come here, dude. Sorry, one moment. No. There you go. Oh. So I wanted to come up. So what you'll notice then is everything is now equal. It has an equal concentration on the outside of the cell as it does on the inside. Now this doesn't mean that movement of the particles stopped. What it means is that the movement will still continue, just in both directions. So what we mean by that is particles will still flow in, but some of them will also flow out. So it'll be nice and equal movement in both directions. That's what equilibrium is. Put this back up here. Diffusion. So diffusion is the movement of molecules from the area of high concentration to an area of low concentration. Now we call this moving against the concentration gradient, meaning that it's going from high to low, from crowded to less crowded, until, like we said, it meets equilibrium. Now, many of the materials that cells need to survive can actually enter the cell by diffusing through the membrane. Now, the nice thing about this is diffusion does not require any energy. It's just a matter of the molecules kind of forcing them to go from more concentrated, so from crowded to less crowded. And that requires zero energy on the cell's part. So because there is no energy that is spent, we do call this passive transport. So it's a type of cell transport that does not require any energy. So diffusion is simply the movement of molecules across the membrane. Sometimes though, there are molecules that want to move from an area of high to low concentration, but either they're too large to pass through the phospholipid bilayer, or they have too much of a charge. They have too much of an ionic charge to move through. Well, in that case, we have what's called facilitated diffusion. And what facilitated diffusion is, it's diffusion, but through a special protein channel, which we can see here. So a molecule that maybe can't go through the membrane for whatever reason, but it still wants to flow from high concentration to low concentration, it'll go through one of these protein channels. Now it's called a protein channel because, well, this tunnel is made out of proteins. And what'll happen is that these are very, very specific. So they'll only let certain molecules through, but it'll allow that molecule free passage back and forth depending on which side has the higher concentration gradient. Let's see, since it still requires no energy, the proteins are just sitting there like a hole or like a little pore in the membrane. It does not require any energy from the cell, so it's still considered passive transport. It's just a matter of transport or diffusion with the aid of a protein channel. Right, Ripley? Yeah. All right, let's talk about osmosis. So osmosis is a term we give to specifically the diffusion of water through a selectively permeable membrane, like a cell membrane. Now, the thing is, sometimes some molecules cannot pass, some solutes cannot pass across the membrane at all, whether it's because the cell just doesn't need to take it in or what. They cannot go from the outside to the inside or inside to the outside. And if they're not in equilibrium, then that solution that the cell is sitting in, we give it one of three terms. And I'll explain these a little bit more in a moment. Now, what these do is they actually call, it's okay, dude. Oh, you don't like osmotic pressure? I know. So this causes osmotic pressure. And what that essentially deals with is if you can't move the solute in order to, if you can't move the solute in order to reach equilibrium, instead you move the water. So what we see at the bottom here, look at the pictures, there is this large circle that represents a solute. Now we clearly have more of it on one side of the barrier than the other. Now it can't go through that barrier, it's much too big. So what happens instead is the water will flow through the barrier to try and dilute the side with the higher concentration. So if you can't move the solute in one direction to lower its concentration, you can move water through in the opposite way in order to dilute the higher concentration, so you can lower it by adding more water solute. So the mass of the solute will stay the same, but you are increasing the volume of the solution. Isotonic solution is actually when we see the concentration equal on both sides. Now, the nice thing about this, we don't have to worry too much about trying to keep the big solutes in equilibrium. The water molecules, just like before, they'll still move back and forth. They'll still move across the membrane, but they'll do so equally in both directions in order to maintain equilibrium. Hypertonic is when you have a higher concentration of solute outside of the cell. So the solution itself has a higher concentration of solute. Now what happens with this is because a solute has a higher concentration on the outside, water actually gets pulled from the cell to the outside of the cell in order to dilute that higher concentration. Since water is leaving the cell, this can cause the cell to shrivel up and possibly die. Hypotonic solution is when there's a lower concentration of solute outside of the cell as opposed to inside. So you have more solute, sorry, you have more solute inside, less solute outside. Again, since the solute particles can't move, then that means water is going to be pulled to the inside of the cell in order to dilute the higher concentration inside. Well, since you're filling it with more and more and more water, it can cause the well to actually expand, bulge out, and even possibly burst. And that's what we talk about when we're mentioning osmotic pressure. So here are some examples. In this case, you have a hypotonic solution. So there's a lot more solutes inside the cell than outside. Water will flow into the cell to try and dilute it, causing the cell to expand, puff out, and possibly burst. Just like if you fill a water balloon. Starts out small, you add water to it, it expands, it gets bigger, and if you keep adding more and more, eventually it'll pop. Let's see. A student a couple years ago gave me a good way to remember that. Hypo sounds like hippo. Hippos are fat. Hypotonic solutions cause the cell to get fat. Isotonic is when there's an equal amount of solutes outside and in, so water is going to flow in and out. It's going to flow in both directions and just stay in equilibrium. Hypertonic is when there's a higher concentration of solute outside the cell, and this is going to cause water to move from the inside to the outside. So water is going to leave the cell in order to try and dilute the outside concentration. This can cause the cell to wither, dehydrate, and even die. All right, let's talk about active transport then. So we said passive transport, there was no energy required by the cell to move solutes or to move material across the membrane. Well, in active transport, you do need energy. Energy is required for this process, and they do it using ATP molecules. Now remember, we discussed when we were talking about the organelles, how the mitochondria breaks apart glucose in order to create energy in the form of ATP. And then we said how ATP goes around and powers everything in the cell. Well, this is one of the things that it would power. There's a few different ways that it'll do this. One way, which is pictured here, is using something called protein pumps. So they're kind of similar to the protein channels, except they use energy to actually kind of open and close. Now, the reason why we need to use energy is because we're not moving things from high to low concentration. So we're not going from crowded to less crowded. We're actually going in the opposite direction. We're going from an area of low concentration to an area of high concentration. So we're moving against the concentration gradient. And the reason why this can occur is if we need to build up a large supply of different ions, such as calcium ions or sodium ions or even potassium ions. We want to build up a supply of those because they could be used to send neural impulses through the cells. Well, if we don't have a large stockpile of them, our cells won't be ready to pass along one of those neural impulses when the time comes. So they want to gather as much as they can and build up a stockpile of it. Now, if you see from here on the picture, this is a protein pump. And notice how it's closed on one side, but it's open here. So the solute, maybe it's a sodium ion, can attach itself to the inside. Once it attaches itself to the inside, ATP will provide energy to the protein. That'll cause the protein to close off the top and open the bottom. Now, when it opens the bottom, that's going to push that sodium ion into the higher concentration within the cell. Then once the ion has left, it reverts back and closes at the bottom and opens at the top. Waits for another ion to fall in there, uses more energy, closes the top, and pushes it in. So that's one way to do active transport, is with pumps. Another way is something called endocytosis. So endocytosis is the process of taking materials into the cell by means of infolding or pockets of the membrane. So what we see here is, this is the outside of the cell, this is the inside. What happens is there's a group of molecules and the cell membrane will actually start bending inward. It'll bend in and then fuse across the top. So start kind of like this, and then it'll close up along the top until it fuses. I guess I should maybe do it like this. So my arms are the membrane. What happens is it'll actually fold inward, and then once it connects like that, it'll fuse surrounding the material, and then that fuse section will break off and it will move into the cell. Now there's two different types of endocytosis based on what's being brought in. If you're taking in liquids or a lot of smaller molecules, we call that penocytosis, like you see here. If it is a larger solid or food molecule, then we'll do phagocytosis. It's the exact same method, it's just we give it different terms based on what is being engulfed and taken into the cell. Exocytosis is the opposite of endocytosis, where in endocytosis, we were folding the membrane around something to bring it into the cell. Endocytosis is the process of releasing stuff to the outside of the cell. So it works exactly like endocytosis, but in reverse. So we have a vesicle or a membrane, or a vesicle or a vacuole already. And what will happen is it'll fuse with the membrane. When it fuses with the membrane, then it starts to open up and it'll push whatever it has to the outside. So here we see the vesicle. It fuses with the membrane. Once it fuses, it then stretches and opens up, and it'll push that material to the outside of the cell. All right. Now let's talk about some additional resources. So first we're going to start off with Science in Pajamas. I have a couple of videos that I made for you guys. One describes the different aspects of passive transport, so cell transport that does not require energy, such as diffusion, facilitated diffusion, and osmosis. And then I have a video on active transport, where we talk about the different types of active transport, which, remember, is cell transport that does require energy. So what happens then is it'll take in we'll describe how it uses energy to run protein pumps, endocytosis, and exocytosis. All right, and then some other videos to kind of help you along. We have the Amoeba Sisters cell transport video. I love these two ladies. They are amazing. They do a great job of breaking the information down so that it's understandable. And they're just fun to watch. Amoeba Sisters again on diffusion, so passive transport. And then we have Amoeba Sisters on osmosis and water potential. So they're also going to talk about hypertonic, hypotonic, and exotonic. And then we have Teacher's Pet active transport video. So it describes the different types of active transport. And then we have Fuse School transport in cells, focusing on active transport again. So I hope that this would help you guys to understand what we've been talking about in the chapter. I hope it helps you to kind of grasp the material a little bit better. I will post a link to the actual Prezi in the description. This way you can have access to the additional resources links directly. But in the meantime, you guys, if you have questions, hit me up in email, Google Classroom, etc. But in the meantime, stay awesome, stay curious, and most importantly, stay safe and healthy. All right, you guys, take care. Bye-bye.